So, as I told you, we established this PXE International Center of Excellence in Research and Clinical Care. So I do see patients with PXE, but I spend most of my time in research with focus on PXE. And also, as I said, we organized this research meeting earlier this week. So what I'd like to do is give you a little summary of where the research on PXE is at this point, what was learned during this recent conference that we had here, and go from there. So there are several different areas of research where PXE is concerned, and we actually know now pretty well the molecular basis of the disease. In other words, what are the changes that follow the genetic changes? So you have the primary change in DNA, but then the downstream, what are the effects that cause mineralization, calcium deposit in those tissues that are affected, specifically skin, eyes, and the vascular system. And I talked about these modifier genes, which is kind of interesting, trying to figure out why some people have more severe disease, some have milder disease, etc. And there are a number of steps that result in calcification, and we are trying to understand those discrete steps in the hopes that we can identify specific pathways, specific points where you can interfere with the pharmacological approaches and the treatment, with the idea of prevention of mineralization. And in fact, we have a number, I'm going to talk a little bit more, we have quite a lot of information on preventing the mineralization. The work was originally done in the mouse models of PXE, and now they are in clinical trials. And then there was a question earlier about the reversal of existing mineral deposits. That's something we are trying to develop new approaches, but as I said, the first question is, if we prevent any new mineralization, how long is it going to take to get rid of the existing mineralization, which is there when the diagnosis is made? Because that's why you initially are diagnosed as PXE, because you have those mineral deposits which give you the problems in the skin, eyes, and the vascular system. Now, I talked about the pre-symptomatic diagnosis, doing in the family before the patients start developing of those signs, and then prevention of the mineralization would be an approach to go. Now, we know now pretty well, so the mutations reside in this gene ABCC6, and the mechanism is such that this ABC, it's a pump. It's expressed in the liver. It's only in the liver, a little bit in the kidney, but primarily in the liver. And it facilitates release of the molecule called ATP. And this ATP is then broken down into these two molecules. And the critical here is the inorganic pyrophosphate, or PPI. And this inorganic pyrophosphate in normal situation is an inhibitor of the mineralization. So it prevents this hydroxyapatite crystal formation. So in other words, sort of like bone structure, you don't get these mineral deposits if you have enough PPI. But now, if this pump doesn't work, you don't have enough ATP, then you don't have enough PPI, and therefore you start getting the calcium depositing these different tissues. So that's the kind of the basic mechanism which is now being refined with the steps and trying to figure out what controls these different conversions and the transports, etc. Now, talking about prevention of ectopic mineralization, or prevention of calcium deposition on those tissues where it shouldn't be, skin, eyes, and vascular system in normal. And there have been a couple of approaches that were all initially developed on the basis of animal models. So we made a knockout mouse which is, we call it PXE mouse because it develops mineralization exactly in the eyes, in the skin, and the vascular system like the patients do. And one of the early studies we looked at was magnesium. How about dietary magnesium? And Mark Leopold is going to follow a little later to talk about the results of a clinical trial that was taking place in Mount Sinai last year. You may have seen or heard there was a idea that because this pyrophosphate, this PPI molecule, it's low. How about putting it back, giving by mouth? And Dr. Varadi is going to talk about that approach shortly. So just drink pyrophosphate and wow, maybe it works. But one of the problems with pyrophosphate is it's metabolized very rapidly, so it's eliminated from the body very rapidly. So we looked in these animal models to bisphosphonates, which are basically stable pyrophosphate analogs. So these have a much longer half-life, and we've been testing whether they work as the pyrophosphate should work. And in fact, there was just recently, I think it came out in October or November, in cardiology literature, a study was done in the Netherlands, feeding people with bisphosphonate, etidronate to be a brand name, and they suggested that there was a little bit improvement in 1 year follow-up period. Now, bisphosphonates are not quite innocuous molecules, so PXE International has taken the position not to recommend at this point bisphosphonate taken that by mouth until we know more about the long-term side effects. And Sharon is coordinating studies to look into that both at the clinical level and use also these mouse models. But then in this meeting we had a couple of days ago, there were a couple of new ideas. One is to use so-called TNAP inhibitors and approach that with gene therapy. In other words, the gene is, you have, PXE patients have this damage, how about putting a good copy of the normal gene in? And so I'll summarize some of these work. One is the TNAP inhibition, and was done in our lab basically with Lily, who's in charge of the animal models, and John Huang with us in the PXE International Center. And so basically the idea here is that, so I told you that this PPI is required, and its synthesis is required from ATP to PPI. But this PPI is normally degraded by another enzyme called TNAP, or tissue non-specific alkaline phosphatase, or cognoscenti. So one way to increase this PPI would inhibit this breakdown, so you get now higher levels of PPI. And how we do this, we actually make two ways. You make a genetic experiment. These are our PXE mice here. They don't have any of this APC6 gene, so they get mineralization in tissues. And then we cross them with a mouse that has reduced, 50% reduced level of this enzyme, which breaks down normally PPI. And then we have normal control mice, and so we generate these mice which have, these are APC6 knockout mice, so these are pure PXE mice, and they have normal levels of this TNAP. This is the gene that makes the TNAP. And we have a mouse which has PXE plus reduced TNAP levels. So is that going to change the mineralization in these mice or not? This just shows the mice look perfectly normal outside, but if you biopsy their muzzle skin, the vibrissae, so the mice have kind of whiskers, as you know, and if you take a biopsy of the whiskers, here's the whisker hair itself. It's surrounded by this blood vessel, and outside is this kind of connective tissue sheath, which becomes mineralized. So it's a skin mineralization. The darker this is, so this is normal, normal, non-mineralized area. This is all calcium deposits. So we can take a biopsy and look by histopathology, like Lionel was talking about histopathology. You look in the skin, you look for the calcium by special stains, and you can actually chemically measure the amount of calcium so you get precise quantitation of the calcium in these mice after various kind of manipulations. And basically, this is if you have reduced TNAP activity in the mice. You get here, this is the calcification. These are mice, PXE mice, normal levels of this TNAP enzyme. If we reduce the TNAP to half, you get less calcification. As you can appreciate semi-quantitatively by this black color here, and you can actually quantitate by calcium assay, so there is a 50% reduction, so you get much less calcium in the skin. And actually, this is genetic experiment, sort of proof of principle, but there is a drug that inhibits this enzyme. And this drug, it has a kind of code name, we don't know what it is, but SPI 425, same thing. If you feed the mice now with this drug, which inhibits this enzyme, which breaks down PPI, you get less mineralization, and you can actually quantitate by calcium assay. So this is a, I think, 4-week treatment of the mouse, so you get much less mineralization developing. So in this kind of, along this line, there are now pharmaceutical companies who are following up this way, and they are trying to develop better and more effective molecules. So hopefully there soon are just pills which will work through inhibition of this TNAP pathway. And that's something probably is going to be in clinical trials within a couple of years. And the gene therapy, so you heard of gene therapy. You just put the normal copy of the gene, and they are often mediated the delivery by these kind of virus type systems. So we've been looking into a possibility of putting the APC6 gene, which is defective in PXE, put a normal copy of that back to the, ultimately into patients, but at this point to the mice, to these PXE mice. And this is one of the works obviously being done in the PXE International Center of Excellence in Research and Clinical Care. And basically, so you make two kinds of viral constructs, but they both have a human ABCC6 gene. So the snippet of human DNA which codes that protein which is missing in the PXE patients. And we make two vectors. One is promoterless, it doesn't have the regulatory elements that are required for the expression, and then we have this kind of viral promoter. So basically, this is inactive, it shouldn't express, but it's a control. And this is an active gene therapy vector, which we hope you put into the cells in culture or in mice, and you start getting the protein, normal human protein being made. And this is just a couple of slides here. So this is the control, this is the promoterless. Here we put those vectors into cells in tissue culture. These are isolated liver cells. And as you know, the ABCC6 is primarily expressed in the liver, so we use liver cells. We look into the expression of this protein, and the more you have this fluorescent green color, the more you make the protein in the culture. And this is different amounts of the vectors, so the more you put the vectors, the more you get cells making the normal human protein. And then we can look how long it lasts. You get one shot, you get lots of protein expression, and then you stop giving more, and you see how long it lasts. And you see this is 1 week, 2 weeks, 4 weeks, 6 weeks, 8 weeks. We still at 8 weeks of times have some of the protein left. Now, that may sound like a short term, but there are ways to modulate this structure so that it's even longer lasting, and maybe you give even more. And this just shows that it goes to the right place, to the right cells and the right location within the cells in the liver. Now, does it do anything to mineralization? That's obviously the question. So if we get more ABCC6, do we get more PPI and therefore inhibit the mineralization? Well, in mouse, yes. What we can do is inject this vector to the mouse that it goes through the tail vein. And this is a control mouse. So this is inactive vector here. You get mineralization, as you see here in the skin of these mice. And then we give them 2 different doses of this viral vector. This is 1 times 10 to the 9th, and this is 4 times more of the virus. And you look this staining, there is absolutely no calcification at all. So introducing this vector, which is active in 2 different concentrations, you get inhibition of the mineralization, and this is quantitation. So this is a wild-type normal mouse. These are the knockout mice without any gene therapy, and then these are the 2 different concentrations. So we are able to actually prevent the mineralization, and the technology for getting liver treatment is being developed in several universities. So this is potentially a pathway to get a kind of permanent type of cure for PXE from early on. So adenovirus gene therapy, we actually shown in the mice now that it is effective up to 8 weeks. And we actually showed that it restored the plasma PPI levels and therefore prevented the ectopic mineralization. We actually also looked into whether it can reverse the existing one, and it did not. So it comes back to the same issue that you asked and highlights the importance of getting early diagnosis, starting treatments as soon as possible the diagnosis is made. But hopefully there are new approaches to get the existing mineralization removed, and we have actually some pretty good ideas being submitted to National Institutes for Health for finding. So this is probably kind of a feasible pathway for gene therapy for PXE, and as I said, there are a number of other diseases, metabolic diseases, which are similarly being tested for viral mediated gene therapy. So this is all kind of work being done in the PXE International Center, and these are all the people that have been participating. Now, one person who is not in this list, but is the key person in the center, is Dr. Koen van de Vedering, who joined us 2 years ago, I think, from the Netherlands Cancer Institute in Amsterdam. And Sharon was telling us in the research meeting, one of the most commonly asked questions is, does exercise improve symptoms with PXE? I guess you've been occasionally wondering yourself that. So Dr. van de Vedering may have some answers. So Koen, why don't you come and show your spiel there? So, and